Hi, welcome to Physionic, or welcome back to Physionic. In today's long-form video, we're going to be discussing and trying to answer the question of how unsaturated fat affects our muscle cells, more specifically in the areas of mitochondrial health and a little bit on insulin signaling or insulin sensitivity. This is a continuation, one of the many continuations off of last week's episode where I discussed the effect that saturated fat had on muscle cells. So this research comes out of the same research group actually, and they decided to extend their research from just looking at saturated fats. Now we're going to also be looking at unsaturated fats. So it's going to kind of drive us down this direction of trying to distinguish between two different types of fats by their molecular structure. Again, I should warn you that if you want a more concise understanding of all this, maybe not all of it, but kind of the highlights, then I'll have short form videos that are going to be coming out on this topic as well. So I'd recommend those over these. However, if you are interested, you are a nerd, you're a geek, you just want to know every little piece of information that you can possibly know on the topic, then this is the video for you. This is probably going to be, just like the last one, about an hour, maybe a little bit longer, because there's a lot of data to get through, and that data comes from a particular paper that I'll introduce to you in just a second. But before we get into that, if you're not familiar with who I am, my name is Nicholas Verhoeven. I'm a PhD candidate in molecular medicine, and some of my research is actually somewhat related to this, in that I am a cell biology researcher, and I'm currently using some of the models that they use in this, or some of the techniques, I suppose I should say, that they use in this particular paper. So, with that introduction out of the way, let's go ahead and jump in, and I will have timestamps and whatnot for you, so if you'd like to skip to different sections. But as usual, with these kind of long-form videos, I really don't recommend that you skip, because every single piece of data is going to be building a story to the end. And then at the end, I'm of course going to put it all together for you in a conclusion. But on top of that, I don't want you to have this expectation that you're going to get so much information out of this paper that you're then going to be able to change your life based off of this one paper. This is going to give us some really interesting clues, but I actually have some more papers that I'm going to be covering that I've already read, that I've already taken notes on, and you can find the notes and this paper linked to this content. But for the future papers, they're going to build off of this paper, and they're going to come from different research groups, so that we can start to kind of create a story of like, oh, okay, this is how I should change my life. So that said, let's go ahead and jump into this paper. All right, so the question that we're going to be examining is, how do saturated fat and unsaturated fat differ in their impact on insulin signaling and mitochondrial health, specifically in muscle? So with that, let's see what we can do. All right, so the first thing is to show you the paper that we're going to be going over. So it's a paper out of the American Journal of Physiology, Endocrinology, Metabolism, and it's called Different Effects of Oleate, which is the unsaturated fat, versus palmitate, which is the saturated fat, on mitochondrial function, apoptosis, which means cell death, and insulin signaling in L6 skeletal muscle, a role of oxidative stress. And it's actually not just going to be a role of oxidative stress, but they're going to be focusing on this as well. Now, L6 skeletal muscle, just so you're aware, these are rat cells. So this is a model of looking at mechanisms. However, just like I said in the other podcast, we're going to see a lot of carryover into human cells, as I'll be presenting in the coming weeks. So don't let this necessarily scare you off, but keep this in context and make sure that you're aware of that. Okay, so a brief kind of introduction. Let me go ahead and remove my image real quick. A brief introduction specifically on mitochondria, because this focuses a lot on mitochondria is that you have healthy functioning mitochondria that help you in the production of cell energy. They're necessary for the production of cell energy if you want to go through what's known as an aerobic metabolism. Now, that said, mitochondria do a lot more than just cell energy. They're also involved in programmed cell death. They're also involved in iron formation, iron cluster formation. There's a lot of different functions, but you don't really need to know all the different functions of mitochondria. You just have to know that you have healthy mitochondria and you have unhealthy mitochondria. And the unhealthy mitochondria tend to have mitochondrial dysfunction. And we'll look and examine a few different data points that look at specifically what that means, mitochondrial dysfunction. Now, in diseases, several different types of diseases, really huge diseases, you have this association with mitochondrial dysfunction. And again, another thing that I mentioned in the other podcast is that, so my research is in mitochondria, but I think a lot of researchers get so caught up in their own research that they start seeing the problem to all diseases is related to the thing that they are studying. And they start thinking that what they are studying is the reason for these diseases. I don't go that far. I think that our body is multifaceted, our cells are multifaceted, and there's a number of contributors. But mitochondria is associated to diabetes, different types of cancers, dementia, and many, many other diseases as well. So is mitochondrial dysfunction the cause of diabetes? Maybe, but it could also just be a contributor, or diabetes could happen and then you have mitochondrial dysfunction and it is simply a consequence. So, but the point is that you have mitochondrial dysfunction that's in the middle of all of this. Excuse me. Okay, so let's look a little bit at how they designed this study, but the number of different experiments that they look at. So the first thing to look at is, well, I suppose I'm cutting off the word there. The first thing that they look at is cells. So these are skeletal muscle cells that are being plated in dishes, cell culture dishes. And this red stuff is media. This is the stuff that we use in the lab as well. And this media feeds the skeletal muscle cells so that they can continue to survive, because obviously they need glucose and they need fats and all those different things so that they can continue to metabolize those to create the different structures within, amino acids, of course, being another critical aspect. Now, the researchers then add either the saturated fat palmitate to the media at varying concentrations, as we'll see, and I'll explain that further. So they'll add it at one concentration and then continue increasing the concentrations in different plates. Or they add the unsaturated fat oleate to the media, or another condition is a combination of the two. And this is going to end up becoming really intriguing because we're going to see some really interesting effects of the combination. Okay, so that said, let's begin looking at the data. And again, I'm going to be breaking this down, and I realize that this can be a little bit overwhelming at times, but hopefully I'll do it justice by explaining things well enough for you. Hopefully I can keep my video up, so sometimes I like to use my hands to illustrate different things, but I also have images here, diagrams, so that you can actually see what's going on. All right, so in this first area, they're looking at DNA. So the DNA that's found there's two different sites for DNA, or our genes, that are held in the nucleus of the muscle cell. So we have a single muscle cell here, and we have skeletal muscle cells are multinucleated, meaning that they have multiple nuclei. And in this area known as the nucleus, that's where our cells contain the vast majority of our DNA, of our genes. However, about 1%, or maybe slightly less than 1% of our DNA is kept in the mitochondrion, or the mitochondria, as they expand across a skeletal muscle cell. So we're not, in this particular data, we're not looking at the nuclear DNA. We're specifically only looking at mitochondrial DNA as a measure of mitochondrial function or dysfunction. So here we're looking at the amount of DNA break frequency. So per X amount of DNA, how many breaks, how much damage is occurring to that DNA. So the higher the value, then the more DNA damage that's occurring specifically to the mitochondrial DNA. All right, so here we've got our three conditions. So remember, they've added palmitate, they've added oleate, or they've added a combination of the two. So, and here's our concentrations. We have a 0.1 millimolar, 0.25 millimolar, and you don't need to know what millimolar is, except that you just know that this is an increase in concentrations from left to right. All right, so here we see that with palmitate, we see an increase in DNA damage. Now, you might be looking at this and thinking, well, all right, it also seems like there's an increase in DNA damage with the palmitate and oleate combination, but that's not the case, at least statistically speaking, and that's most likely because these are known as error bars, and the error bar is really wide in this situation. So it's most likely not significant for this condition. However, it is significant for this condition, meaning that there's only an increase in DNA break frequency with the addition of palmitate or the saturated fat. And then, of course, we see this effect that continues to grow, of course, with a higher concentration and even higher concentration. And you can see that the combination of palmitate and oleate, so saturated and unsaturated fats added at the same time, seem to be non-significant. So they would be, by comparison, the same as the addition of the unsaturated fat alone. Now, this is going to be really, really interesting. I'm going to tell you this right from the get-go, because as I go into some of these other studies that we're going to see over the coming weeks, in humans, just keep this in mind, because I have a theory as to what might be happening here. And of course, that may change as I look into more data. But at the time that I'm recording this, I have an idea as to what's going on here and how we can translate this into our habits, our nutrition habits. But for the time being, just knowing that saturated fat alone increases DNA break frequency. And we don't see that there's it's a stepwise increase up to a point, and then it stays about stable. All right, another measure of mitochondrial function is to look at the cellular ATP level. So ATP is adenosine triphosphate. It is a molecule that's used for cell energy. I'm not going to go into too much of the more detail than that, other than you have what's known as ATP hydrolysis, and then you get the release of energy. But this ATP largely comes from the mitochondria. So, changes in ATP concentration within the cell would be potentially, most likely, a reflection of ATP generation from the mitochondria. And what we find here is, again, we've got our same conditions, and you're going to see these conditions really throughout. Cellular ATP levels seem to stay normal or elevated when you have the addition of the oleate, in this condition right here, or the combination, but you start seeing a decrease with the palmitate. So until it gets pretty significant. It would have been nice for them to have some statistics running across these different conditions, as opposed to just staying within one grouping, one concentration of fat. So here they're comparing the palmitate compared to the oleate or the mixture, but it would have been nice if they had, like, let's say, this oleate at 0.75 millimolar and compared that against the 0.1, because I think most likely, I don't know this for a fact, but I would imagine that you would see a significant decrease as well in the oleate condition, which would be interesting to try to figure out why. But the point is, no matter what, the saturated fat palmitate seem to decrease ATP concentration. This is actually something I've been discussing in the lab, something I've been investigating myself in the lab, and I have some interesting theories that I'll probably end up releasing some content on that as well in probably a month or so, because I have a backlog of other content that I'm trying to release to make sure that we get to answers related to this topic first. And then here we've got oxidative stress. So oxidative stress is, again, I'm going to be simplifying things because I don't want to get too off track here, but you have within the mitochondria, and you have these proteins known as the oxidative phosphorylation proteins. And these, the big picture here is that they allow for the production of ATP. So the very same ATP that we measured here in the entire cell, this ATP synthase protein allows for the production of ATP. Now there's a series of chemical reactions that have to happen between these other OXPHOS proteins, these four complexes, for this protein to function. I'm not going to go into the details with that, but the point is that they have to be functioning, they have to be constantly reacting with one another. So, that said, when that happens, sometimes you get the slippage of electrons, and that ultimately leads to the production of molecules known as reactive oxygen species. These are unstable molecules that, if they interact with other molecules within the cells, like functioning proteins, lipids, carbohydrates, things like that, that actually serve a function, that have been adapted to serve a function within the cell, they can start to tear away at the chemical structure of those molecules, and thereby making them less functional or create more damage. Now, this is all known as oxidative stress. So the higher the oxidative stress, typically the worse the cell is in terms of their health, because there's more damage that's occurring just kind of at a random pace across the cell that the cell then has to fix. And I said this on the last podcast as well, you don't want to eliminate reactive oxygen species completely, because they do actually serve a healthy, functional role inside the cell as well. And I realize that sounds really confusing, considering that they're unstable and that they tear away from other molecules. But a small amount of reactive oxygen species is actually quite beneficial for a number of different processes. One example in muscle cells is that they allow for greater growth signaling. So up to a point, and then once they hit a threshold, the idea is that if you go too high with them, you have too much oxidative stress, that then you ultimately damage the cell too much. Okay, so here we've got our measure of oxidative stress, and we can see that there's an increase in oxidative stress specifically, and it's almost stepwise, with the increase in concentration in, you guessed it, palmitate, so saturated fat, and yet we don't see that with the combination or alone with the unsaturated fats. We'll be revisiting this because it's going to be important as we go into the mechanisms. And I think this is the final piece of data specifically on cell health and mitochondrial health. I could be wrong about that though. This is the viability of the cells. So how many cells are still alive after 24 hours, let's say, of treatment of these fats to the cells. And what we find, and this is same as what they did in the last paper that they released on this, specifically looking at just palmitate. Here we see that a small level of free fatty acids, or fats, which are these, does not seem to affect the cell's ability to survive. However, if you start increasing the concentration, there is a decrease in the number of cells that survive. Okay, now I have a few qualms here, as I did with the last paper. This paper is not perfect. There are some issues here. So, one is that you don't see a step, like, it doesn't seem like there's a stepwise decrease. At least statistically, we see that the half molar level of these fats seem to be non-statistically significant in terms of creating cell death. So again, if this decreases, that means there's more cell death. And again, the same, so we've got this initial decrease and then we have non-statistically significant, and then we have again statistically significant, and here we have it statistically significant again. So this is the data. It's not, I mean, I'm glad the researchers showed this because they have to show it, like this is how it came out, so this is just the way it was. But it could be just through experimental error. It could just be that's just the way that things turned out, but maybe if they'd increased the number of samples that they'd had over time, then they would have started to see something significant here. Okay, that's fine. That's not really a problem. The point is that it's just a little bit confusing in terms of trying to interpret this. So you should probably just interpret it the way that you see it, if you buy the data or if you don't buy the data. Another issue here that I find is that this condition right here, it's I suppose what they did is they're doing a comparison just between the palmitate and the combination treatment of oleate and palmitate. But this one right here really seems like, at least visually, that it should be statistically significant, especially with these error bars, and yet it isn't. So I'm wondering if the statistics were improperly administered here. In that, typically what you would want to do is an ANOVA statistical analysis, where you're comparing all three, so just looking for a difference, and then you do what's known as a post hoc analysis, where you try to figure out, okay, well, what is the true significant difference between, let's say, one condition versus another condition. And I don't know. I think I remember looking at the statistics, and it seemed like they said that they did an ANOVA, but it just looking at it, it just doesn't seem that way. But if you're a statistician, certainly you know more about this than I do, even though I do enjoy statistics. So if you have any theories as to why this is the case, then please let me know. I'd be interested. But if we take the data as it is, the point is that generally, we'll just say generally, we see a decrease in cell viability, so more cell death with the palmitate condition, and we do not see that with the other two conditions. Okay, now this looks confusing. I get that. Here we're looking at two specific proteins. Let me explain those proteins, and then I'm going to explain to you how to look at this. Let me get rid of my visual because apparently I'm obscuring things. Okay, so again, we've got our increasing concentrations of each fat. We've got palmitate alone here. We've got oleate alone here. We've got our oleate and palmitate combination. Here we've got our control condition where none of the fats were added. So this is just the regular media to the cells without any added fats. And they're measuring the amount of PGC1 alpha and TFAM. So these are transcription factors. Let's not get into the weeds about transcription factors, but the point is that these two are related to mitochondria, specifically to the DNA of mitochondria. So this means that the more of these that you have, typically the greater the biogenesis of mitochondria. Let me say that differently. The more of these two proteins, typically the more mitochondria that you have, or the more creation of mitochondria that you have. So if you want to have a lot more mitochondria, which is typically seen as a healthy state, there may be exceptions, but typically seen as a healthy state, then you would want more of PGC1 alpha for sure, and you would certainly probably want more of TFAM as well. I realize I'm not really explaining what these two are, but getting into the idea of transcription factors and activation of promoters and all that stuff is getting into the weeds. And it's not information that you absolutely need, but certainly look them up if you would like, and you'll learn plenty about them because there's a ton of research on both of these proteins. Now, how to read this? Well, if you don't want to read this, you can look at these graphs, which are literally just the measurements of these smudges. So they're in graphical form. So these two correspond to all of this. So it's not like they're separate things. But if you do want to know how to read these, so these are protein levels. And here we have a condition with the control, and we've got PGC1 alpha all the way across here. We've got TFAM all the way across here. And this is what's known as a loading control. So a loading control is a way, and I've mentioned this in other podcasts as well, but it bears repeating because you may not have gone through this yourself with the other podcasts. So a loading control is, for this experiment, you want to know if the experimenters, the researchers, are adding the same amount of total protein to the experiment condition. So when they do this experiment for the control, are they adding a lot more total control and therefore they're getting increases in TFAM and PGC1 alpha? Or is the total protein that's being added to the experiment the exact same across conditions, and it just so happens that there's lower levels of PGC1 alpha and TFAM in these conditions? This is vital information because it could be that you've got normal levels here, and then for these two conditions with no PGC1 alpha, they just had they just loaded very low levels of total protein. That would then indicate, well, what if you raise this up to the normal levels, what you see here? Would you start seeing activity here? Would you start seeing protein levels? So this is vital information. You should have a loading control of some sort. And actin is a very popular one because it's a protein that doesn't change much in its total amounts, regardless of what conditions, what kind of things that you add to the cells. Actin stays extremely stable. All right, so hopefully I did some justice to explain that. So now we're comparing, so actin levels are largely the same. So you're looking at the darkness and the width of the smudge. Largely is a little bit of variability, but when you actually end up quantifying it, it'll give you a number, and then you can normalize that based off of what you're seeing up here. So in the control condition, now that we've explained that, in the control condition, PGC1 alpha, we see that there's a good amount. TFAM, we see there's a good amount, and actin is relatively normal. Then with increasing concentrations, you start seeing, okay, PGC1 alpha may be normal, although here, when they're actually looking at the bar graph, you see that PGC1 alpha seems to be slightly reduced with the palmitate condition. And it's hard to see that, right? But at least when they actually do the quantification, which is certainly more exact than our eyes, unless we're talking about some extremes like we're here, then we typically believe the quantification over our very subtle differences in our eyes. All right, so here we see that there's apparently a decrease, and then certainly the most evident is obviously as you're getting to about half a millimolar to 1 molar, half a millimolar to 1 millimolar, excuse me. Then you're getting to see these decreases in TFAM, especially at 0.75 and 1, and then at half all the way up to 1 for PGC1 alpha, it's essentially gone. And we see that reflected here as well. So this is the same here, and this is for TFAM. So you see a kind of a stepwise decrease. Now this is all on palmitate. What happens when you just look at unsaturated fats? This is not good. This is not what you typically want to see. These are overblown Western blots. This loading control should have more separation between them. And I'm going to show you an example of a terrible example of this later on. You typically don't want to see this. I mean, is this believable? Yeah, I guess it is. But typically you want things to be a lot cleaner. So actin levels seem like they seem normal. And here we've got some variability, right? But again, with this quantification, we're seeing that there aren't any huge changes in PGC1 alpha, at least by comparison of, let's say, this condition to this condition. We see that there is PGC1 alpha, and here we see that there's no PGC1 alpha. And the same with the mixture. It certainly seems darker, and maybe there's slight differences between these two conditions. It's a lot of kind of mental thought, like thinking that you have to go through to be able to like take this into consideration, then compare to this, then compare to this, then compare to this. But I think the most striking differences are certainly if you compare either one of these conditions to the palmitate condition, you see that there's a complete loss of PGC1 alpha and starting to see dramatic decreases in TFAM. A lot of interpretation goes into this, and you usually have to sit here and really study it hard to see what problems there might be. And this is not great, but the rest of it looks okay. Typically, when I take images of this kind of stuff, I try to do it at a lower saturation because, well, I won't go into that. Again, I'm going to go on a tangent otherwise. All right, so this is all in mitochondria, or more measures of mitochondrial function and biogenesis and things like that. Now I'd like to switch gears to looking at a little bit more on insulin signaling, because I know a lot of people are interested in that. And that's something else that they cover by extension to something that they measured in the other video that I released. So just to cover how insulin signaling functions, so you have the release of insulin from the pancreas, so it's insulin molecule here. Then it will interact with the insulin receptor, which then is known dimerizes. So you have two sections that end up coming together, and they kind of hug with one another. Then there's a ton of molecular aspects that happen here. Again, not going to go into those, known as autophosphorylation, and then the recruitment of different scaffolding proteins and whatnot. But the point is that you have these two proteins that come together once, and then insulin binds, then you have them come together. And then you have this protein known as IRS, which will then get recruited to the insulin receptor. There it will be phosphorylated, which means that in this situation, we're talking about it in an activation sense. So IRS protein, this is all within the cell now. So insulin was outside of the cell, and it stays outside of the cell. Then inside the cell, you have a bunch of molecular changes because these two come together. And you have an inner portion, so an intramembrane section. So this is intermembrane. This is extracellular. And you have an intramembrane section or intracellular section, which then also comes together. This recruits IRS. You have an activation of the IRS protein by adding a phosphate molecule to it. And I'm giving you some details that you may not necessarily need to know. Just know that IRS gets activated. And then you have a series of other activities that happen that I've completely just cut out because I don't want your eyes to glaze over. Excuse me. And then from there, you have the eventual phosphorylation of AKT. And this is the protein that we're going to be most concerned with, because the phosphorylation or activation, which it can be phosphorylated to inactivated as well, but in this situation, we're looking at activation. So the activation of AKT then leads to the recruitment of these GLUT transporters, which are blood sugar transporters, which are inside the cell. And then once they're promoted, they get promoted up to the cell surface, where they integrate themselves into the cell surface, kind of creating pores for the blood sugar then to enter. So this then drops blood sugar. And that is the insulin cascade in a very simplistic molecular sense. Now, let's look at that. All right, let me throw my image up here, since you can tell that glucose comes in. All right, so here again, we're looking at these phosphorylation. We're looking at the protein levels. So here, this is the same thing. We've got our control, we've got our increase in concentrations of our fats, palmitate, oleate, unsaturated, and the combination of the two. However, in this situation, because they're measuring insulin, right, insulin signaling, they have to add insulin. So they're either adding or not adding insulin to the media and seeing what happens to the cells. Here we're looking at a measure of phosphorylated AKT, so right here, so with the phosphate, so activated AKT, and the total AKT, which is just the total amount of this protein, not necessarily the activated, just total amounts. And then we've got our loading control as the actin. So the actin looks really good here. This is more so what I was talking about. In this last one, it was I did not like that because it was all kind of blown together, kind of thrown together. It kind of they kept developing it and saturated, and this is not the way that you typically report this kind of stuff. This is definitely the way that you report this kind of stuff, where you can see the individual marks for each condition. However, hopefully you can see what I'm talking about. What is this? This is, let me see if I can zoom in here just for a second. If you look at that. This is horrible. This is so bad. This is just like this is like a snake. It's all put together, like it's all just blown together across the conditions. This is not acceptable. This is not good. Doesn't mean that the paper's bad or anything. No, it doesn't necessarily mean that, but it's they should have done a better job here. All right, so anyway, here we're just gonna have to make some assumptions that the total AKT was normal. I don't see any indications that it's not normal, but this is just not great. Not a great way to report it. Anyway, all right, so we've got our total AKT, and you can see the palmitate, oleate, and the combination mixture seem relatively the same. Maybe here there's a slight decrease, but it doesn't seem, if you end up looking at the ratio, which is what we're more interested in, the ratio of phosphorylation of AKT, so the activation of AKT to total AKT, that's what we're more interested in. And what we find is here, again, you see these dark bands when you're using kind of smaller amounts of palmitate, and then you get to roughly about the same time that you saw loss in PGC1 alpha for the mitochondria, you see a loss in the phosphorylation of AKT. And do we see that when we're looking at the oleate condition? No, we don't. And we don't see that with the combination of the two either. So this is again giving us further proof that there's a saturated fat or palmitate specific effect here that's going on. Now, that said, in the last paper that they released that I covered last week, they covered IRS, they looked at GLUT transporter, they looked at blood sugar coming in. This is the only measure that they used for this paper. I would have definitely liked to have seen more than just this one measure of insulin signaling. So not all that great. However, I'm glad that they showed the actin levels, which look really, really positive. Okay, so now let's look at some of the mechanisms. Let's explain what is happening here. They do a series of different experiments going back to some of their previous experiments, but using a series of different inhibitors. So to give you a bit of background on this, because we're going to be introduced to a few of these players, let me go ahead and, there's nothing over here, as you can see. All right, so I'll keep myself there in case I end up using my hands to illustrate stuff. So here we've got our mitochondria, and we saw that you have some production of the reactive oxygen species, right? So the ROS. So the production of ROS, if it gets to be overabundant, then you get an indirect activation of a protein known as J and K. And if you watched the last video, you know that I kind of call this junk, just because it's missing the one letter. But J and K is a stress protein. It does a lot of other things as well, but it's also known as a stress protein. So it's an indication, if it gets activated by phosphorylation, that it is a stress signal within the cell, or one of them at least. And when it's activated, then it leads to a series of other changes within the cell, molecular changes, kind of like what we were talking about with insulin signaling. And this then leads to the mitochondria essentially dying. And this then leads to another protein known as caspase 3 to be cleaved. So the death of mitochondria lead to a series of molecular changes, again, that I've opted not to show you, because if I show you this kind of stuff, truly, it would be just so complex that you'd have arrows going everywhere. It would be all kinds of stuff going on. And I'm trying to spare you from, and I'm also trying to keep my retention somewhat for these videos so that I could show you the full breadth of molecular biology one time, but it's just a real cluster. It's too much. So anyway, mitochondria end up dying, and then you get the cleavage of caspase 3. What does that mean? It means that the caspase 3 gets cut. And the reason why this matters is because this is in its inactive form when it's uncleaved, when it's in its total form. And then the cleaved caspase becomes activated. Now, what does caspase do? Well, it goes around to the other proteins within the cell, and it cuts them. It's known as a protease. So it starts to destroy other proteins within the cell. This leads to significant damage within the cell. And presumably, if there's too much damage, then the cell ends up opting for cell death. So we're going to look at ROS, we're going to look at J&K, we're going to look at caspase 3. So that's why I introduced those. All right, so the first one is J&K. Remember, the phosphorylated version is the activated version. And that doesn't mean that you're never going to have any activated version in cells, because J&K does other things as well. But in this situation, if you see a significant rise in J&K, that may not be a great sign, depending on some of the downstream effects. So here we've got our control. And again, we're just looking at palmitate, our oleate, and our mixture. Here we've got a phosphorylated version. These protein levels are all phosphorylated. If you don't care about that, you can just look at this diagram right here. And I suppose I should get rid of my... There we go. Now you can see it better. So here's phosphorylated J&K, which is also known as SAPK. And here we've got our total levels. Okay, let me give you the face. This is, again, you know what I'm going to say. And here we've got our actin, which is kind of overblown here. This is okay, and this is somewhat okay as well. I believe that these are all the same, but it's just like for the optics, you typically want to have cleaner looks on all this stuff. So I imagine the reason why they blow these out so much is, I don't rationally, it doesn't really make a whole lot of sense, but really, or at least from a scientific standpoint, because there are ways of getting around this. But I think the reason why they're blowing these things out is because they need so much total J&K to show so much total J&K to start showing the amount of phosphorylated J&K. Because you can see these are much lighter than these. So they have to, if they had these be represented at this level, you would see nothing here because it would be just blank. But they have to keep developing and keep saturating these over and over and over again until they start detecting some signal for these, because the antibodies and the total amounts of these are much lower than the total amounts of the total J&K. So hopefully that made sense. But there are ways that you can get around that, but I don't know. Some researchers just don't do it. Whatever. The point is, here we've got our control, so everything is being compared. All of these conditions are being compared to control. And we see that palmitate, again, you kind of have to squint unless you end up looking at the graphics. Palmitate, here it's pretty obvious. So the 1 millimolar level, you start seeing really a much greater phosphorylation of J&K compared to the control. And here, again, with oleate or with the mixture, they're pretty mild. Sure, you see some phosphorylation because it's there, but it's nothing overly extreme compared to the control. And that's what we find here when we look at the graphics. So the graphs, again, show that only the palmitate increases the phosphorylation of J&K. We're going to be returning to that in just a minute. To look at caspase cleavage, so here we've got our full caspase 3, which is the inactive version, and then we've got our cleaved version, and then we've got our actin. This is really nice right here. The actin looks good. All right, so with palmitate, again, right around that mark, right around the 0.5 0.75, 1. This is more following the TFAM that we saw. We started seeing decreases at 0.75. So the cleaved version is, or I'm sorry, the full length version, which they have a misspelling here. The full length version is roughly the same. So you start seeing this, normal levels again by comparison to control. If you compare all these to control, and even these compared to control aren't that bad. But then once you get to higher levels of palmitate, then you start seeing the disappearance of this caspase 3. And why is that? Because it's being cleaved. It's being cut to its cleaved version. So the cleaved version doesn't seem to appear much, maybe a teeny tiny bit in the oleate and the mixture. But overall, the cleaved version is very highly regulated, and it's even a little bit higher. Well, maybe actually by comparison to control, it's not. So we'll leave these alone. These are fine. But here you start seeing an increase, and here you really start seeing an increase in the cleaved caspase 3, which again, these end up going through the cells and chomping away at all different proteins, start destroying them, damaging them. Suppose I can put this back up. Oh, of course I can't. Okay, let me introduce something new now. So all these mechanisms that I've explained to you up to this point, I discussed them in the previous video. However, now I'm introducing something new, and this is going to be important because this is something that I'm going to be bringing up in the future, and this is going to end up becoming a pretty important aspect. So I'm going to explain to you the molecular workings of this. Don't be thrown off by this. It's not going to be this complicated. And this is the visual that I've created to represent this. So ultimately, I'm going to switch to this so your eyes don't glaze over looking at this. All right, so now we're introducing a molecule known as a ceramide. A ceramide is a combination of a fatty acid, also known as a fat molecule, which is what we've been looking at, the free fatty acids, which are the palmitates, the oleates, and the combination of the two. And typically what happens is with saturated fat, although I believe it's also possible with unsaturated fat, but saturated fat then gets combined through some enzymatic reactions, gets combined with this molecule known as a sphingosine. So the saturated fat and the sphingosine will be combined into what's known as a ceramide, which is a combination of the two molecules, fatty acid, sphingosine, making ceramide. So here I've got this quote-unquote sphingosine. Here I've got the fatty acid, and it ends up forming what we're going to look at it as the ceramide. So if you see this image, just think ceramide. All right, I'll explain to you what that all means in just a little bit. But the point is that you get the formation of these ceramides. That's all you need to know. Okay, so here what they've done is not related to the fats. Here they just want to show what happens when ceramides get added to the cells. Now I just realized that this does not have a y-axis. Let me see real quick. Okay. I'm looking at my notes real quick. Okay, yes. So this is I wish I'd added one more piece of data. That's all right. I can just tell you about it. Or you can just hop on over to the notes as well, which will be supplied for you. All right, so here what they're doing is they're looking at cell death. So this is what the y-axis is. So or cell survival, I suppose I should say. So the same as cell viability that we looked at earlier. And here they're adding increasing levels of, this is not the fats that we've been looking at. This is a measure of the amount of ceramides that they're adding. So the molecule that we were just talking about, this right here. So with increasing concentrations of ceramides added to the cells, they're pretty robust for quite a while. And then at 100, that looks what? Not millimolar, but micromolar. at 100 micromolar of ceramides added, you start seeing significant cell death. Okay, that doesn't mean a whole lot. They're adding a decent amount of this stuff, so clearly it's having an effect. However, what the other piece of data that I wish that I had added, because I completely looked it over, but they also do the same experiment, but with this ceramide, but with the looking at reactive oxygen species production. And what they find is that at 50 micromolar, that you see an increase in reactive oxygen species with the addition of this ceramide. Now, just as a control, they added a ceramide mimic, and this ceramide mimic does not have the same metabolic activity that ceramides do. So it looks very similar to that structure, to this molecular structure right here, but there's some molecular alterations to it that end up making it less metabolically active. And what we find is that when they add it, there's no effect. And the same was true with the reactive oxygen species. All right, now, that said, now let's take that information and let's try to bridge that information with the information that we've gotten from the palmitate specifically. Palmitate has had all these differential effects specifically to the cell, from mitochondria to insulin sensitivity, or insulin signaling, I should say. So here we've got our palmitate. We're looking at the number of cells that survive. Same experiment that we saw earlier, but they're removing the oleate, they're removing the combination of the two, so palmitate, and they still see, again, a decrease in the cell survival that they saw in the other data as well. However, now they've added this FB1. Let me quick look up it's like fumonisin B1. All right, so fumonisin B1 is a drug, a molecule that will inhibit the conversion of saturated fat with sphingosine into ceramides. So there's an enzyme known as ceramide synthase that is enzymatically responsible for the combination of these two molecules. So here they've added this drug, which is a mucomycin, I think. It's a mucotoxin, meaning that it comes from a fungi. I always like debate in my head like how I should say that, fungi. But I remember I had an interview with a friend of mine that is up on the channel, and she studies fungi, and so it's this hour-long interview, and like the whole time I'm like in my head, I'm like, do I say fungi? Do I say fungi? I think I'm starting to opt more towards fungi, because fungi doesn't sound quite right. It doesn't, fungi aren't the greatest. They're not very fun guys. So I'm going fungi. So fungi can release this toxin. So they end up using this toxin on the cells, and what it specifically does is it inhibits the production of the ceramides, so it inhibits ceramide synthase. So in here we see that when they do that, they recover the effect from palmitate. So what does this mean? It means that it implicates palmitate in the production of ceramides. And what those ceramides do, I'll explain to you right now. Okay, so ceramides, there's a number of different functions that they have. But if they are overproduced within the cells, they can get into the membranes of the cells. I guess I'll go full screen real quick. They get into the membranes of the cells. They can also get in the way, so they can attach to molecules within the cells, different proteins, as we're about to see, like AKT, for example, and inhibit the interaction of other proteins, signaling proteins, like the ones that we discussed, IRS, or the GLUT translocation, all that. So AKT has to propagate that signal. Once it's activated, it needs to send a signal further down into the cells. So As such, if you have something that's blocking that interaction of the other proteins to AKT, then the signal can't continue on, even if AKT is active. So ceramides are implicated in some of that, and I'll be touching a lot more on that signaling in the coming content. So just for now, though, know that the overproduction of ceramides are not wanted. All right, so they've implicated at this point ceramides in the cell death pathway, at least, and palmitate. Stopping the production of ceramides protects the cells from palmitate-induced cell death. Okay, so here now they're doing the same, a similar look, but they're looking at the AKT levels or phospho-AKT levels. Oh, look at that. Look how beautiful these are. Total AKT levels look so nice here. This is exquisite. This is very nice. I wish that they had done this before. But here we've got our control plus or minus insulin addition with palmitate. And then they've added these two drugs. So I've already introduced FB1 to you, which is the mucotoxin, which eliminates or stops the production of ceramides. But beyond that, they also add NAC, which is an antioxidant or a precursor to antioxidant, or a precursor to the antioxidant enzyme glutathione. So antioxidants eliminate reactive oxygen species that we talked about earlier. So as such, that means that with the addition of this, so let's back up here. With the control, you see that there's still phosphorylation of AKT. Everything looks good. Obviously you're not going to get any phosphorylation of AKT without insulin stimulation. So with stimulation, you see phosphorylation. This is beautiful. This is really nice. Then here we've got, again, no stimulation, so shouldn't have anything. And here we see the addition of insulin plus the palmitate. You see that there's a dampening of this effect right here. All right, all well and good. Now, when we have palmitate, which should look like this, and we add the oxidative stress eliminator, the antioxidant, NAC, we see a rebound in the phosphorylation of AKT. Maybe not quite to the same as control. That's up to your judge of an eye, but I think we can agree that it definitely increases from here. Then, separately, with the same condition, palmitate added, they add this fumonisin B1. Then you see a rebound in the phosphorylation of AKT again. So through two separate pathways, we're able to recover the phosphorylation of AKT. This is really interesting because it will tell us things in the future. Now, looking at this with SP, so they're doing the same experiment here, but with SP, this is a inhibitor of J and K, so that protein that I was showing you earlier, right here. Phosphorylation of J and K is a stress enzyme. So here we've got, again, this is the same experiment as here. This is the same experiment as here. And again, we see this decrease in the phosphorylation of AKT, all well and good. These look pretty good. And then with the blockage or the inhibition of the phosphorylation or the activation of J and K, then you see a rebound in the activation of AKT. Very cool to see. So we've got three different mechanisms now by which we can recover the phosphorylation of AKT, so insulin signaling, by this one measure, just to be clear, just by this one measure. And then here we've got the caspase, which is, again, that protein that I told you about when it's cleaved, that you get the damage, the mass damage that occurs within the cell and can potentially lead to cell death. Again, control, we see high levels of caspase 3, which is, again, the total caspase 3. This is not the cleaved caspase 3, which is the activated form. This is the quote-unquote inactivated form. And what we find is palmitate, we see a reduction in caspase 3, presumably because it gets cleaved into its cleaved form. And then with the inhibition of J and K inhibition, or J and K, right? J and K has to lead to molecular effects of the mitochondria. Then you get the cleavage of caspase 3. So if they inhibit this, they're going to be inhibiting all this other signaling that's occurring. Excuse me. I'm an hour in. my voice is starting to go. Okay. All right. Now, there are two problems here. There are two problems when it comes to looking at this data. I'll go ahead and mention this one here first. First, they should have had cleaved caspase. To me, this is kind of useless. This doesn't mean anything to me. Just showing, for us to, let me see if I can find the caspase data again. There it is. Okay, so here they actually showed that if you see a decrease in caspase 3, full-length caspase 3, that you see an increase in cleaved caspase. I think what they're trying to do is you saw it earlier, so can you just believe us that this is what's happening here, that you see a decrease in full caspase 3, therefore you presumably see an increase in cleaved caspase? I say, nay, I will not believe you. You must show it. So here, if I saw this data, I'd just throw this out. This doesn't mean a whole lot to me. Although the actin looks very nice, which brings us to the second problem. The actin looks really nice here, and it looks quite nice here. Why isn't it here? This is total AKT. This looks beautiful, but you still need to have a total loading control. This is a little bit less of an issue because you have the total levels, but you should still have it. This is just to the point where this is just data, but it's kind of pointless data at this point because you're not adding the cleaved levels. So just wanted to add those critiques because it's important. And the final piece of data is to look at this oxidative stress inhibitor, or this antioxidant, so NAC, which is the one that we looked at here, and looking at the TFAM levels as well as the PGC1 alpha levels. And what we find here, we've got our control, here we've got our TFAM levels with lower levels of palmitate than an addition and greater levels of palmitate, and it seems to have a protective effect by comparison to when you just have palmitate alone, or what we saw earlier as well. Now the same for PGC1 alpha, we see that it also seems to have a protective effect where it increases the levels of PGC1 alpha. So, well, I'll get to it in the conclusions. Let's go into the conclusions, then I'll be presenting to you some of what we can take away from this. Okay, so in conclusion, this means that palmitate, the saturated fat, increases mitochondrial DNA damage, oxidative stress, and general mitochondrial dysfunction in isolated muscle cells, as we saw in this data as well as the last paper that we covered. It also may reduce insulin signaling, but they need more measures. AKT is a really powerful one. It's a great one, but I think that they should have done at least one or two more measures like they did in the other paper. However, consuming oleate, the unsaturated fat, eliminates these effects, especially if in isolation, but also when combined with saturated fat. Now, the mechanisms, I didn't add this to the conclusion. The mechanisms, let me go ahead and cut to me real quick. Now, the mechanisms are multifaceted. That's one of the really intriguing aspects of this. Is it's not just oxidative stress. I mean, they have that in the title, that it's an oxidative stress mechanism. And yes, it certainly is a major contributing factor. Maybe it's the first contributing factor, that an increase in oxidative stress may lead to increases in the phosphorylation of J and K, which then leads to more mitochondria going towards cell death, or going through mitochondrial death, which eventually leads to cell death. And then you get the activation of the caspases, which then do a bunch of damage to the cell, therefore also leading the cell to succumb to cell death. Now, so it's oxidative stress, it's the phosphorylation of J and K, but we also talked about ceramides. And this is something that I'm going to be pursuing in the upcoming content in the next several weeks. We're also going to be looking at ceramides. And then, so because they did that with the inhibition of ceramide synthesis, but they also did it with the inhibition of antioxidants. And both of those had pretty significant effects, actually extremely significant effects in protecting the cell from cell death, but also from mitochondrial damage and rebound the health of the mitochondria, at least based off of the measures that they had with TFAM and PGC1 alpha. So, we're learning more and more. This is, of course, just in cells, just in muscle cells. It's a model of muscle cells. So they are muscle cells, but they're not from humans. So what's going to happen when we look at the effect in humans? Like not just cells, but also in humans. Well, let's find out. So in the next several long-form videos, that's exactly what we're going to be investigating. And we're going to see if the effects that we see here are also true for the effects that we see in humans. One more thing I'd like to add here is that people are commenting. So people are adding comments of, like, well, how could this be? We've been eating saturated fat for such a long time, and that's fair enough. That's a decent argument. Sure, we've been eating saturated fat. Some people have been rebutting, like, well, we haven't lived as long when we are consuming large amounts of saturated fat. There's all kinds of like rebuttals and whatnot that you could have. I think what might be happening here is, if this data ends up being true, that we don't exclusively eat saturated fat. I would bet, just based off of the last two studies that we've looked at, and I will be showing you again more studies. I keep teasing that because that's important. I would bet that it seems like unsaturated fats have a protective effect on, as we saw with the conclusion of the data, in every measure. Unsaturated fats seem to protect against the drastic effects of saturated fats. So what are the odds that you only eat saturated fat? It's pretty low. I mean, we have unsaturated fats in our other consumption. Our body stores certain levels of unsaturated fats, like omega-3s, for example. So what if the combination of consumption of unsaturated fats and saturated fats leads to a protective effect on the body, so that saturated fats' negative effects are at least drastically diminished by the concomitant consumption of unsaturated fats? So what if the unsaturated fats essentially are masking the effects of saturated fats? But if a person were to completely just eat saturated fats, and also I think maybe a stipulation of that would be they'd have to deplete their unsaturated fat levels in their body, that this would then lead to drastically negative effects of saturated fats. I'm just floating that idea out there. I'm not convinced. I'm not saying that that's definitely what's happening, but that's a pretty cool thought, I gotta admit. And I'm going to be pursuing that. I'm going to be showing you more on that. So, all right, I'm done. I'm done with everything. If you made it to this point, big kudos. It's been an hour. It's been over an hour. So congratulations. And I'll catch you next time. Have a good one. See ya.